So T-cell lymphoma on its own is already very difficult to diagnose, but in cases of cytology specimens where maybe flow cytometry wasn't sent, or if you have suspicion for a T-cell lymphoma, this can be a useful surrogate for TCR clonality testing. And the way that this dual stain works is it utilizes an immunostain for the TRBC1, which is the constant region of the T cell receptor beta chain. So most T cells are alpha beta T cells, and in their maturation, either a copy of TRBC1 or TRBC2 will be rearranged and form the T cell receptor complex. So these can be separated by immunohistochemistry, but for flow cytometry, our TRBC1 and TRBC2 antibody exists. But for IHC, we just have TRBC1, so we combine this with CD3. And in our stain, the brown stains TRBC1 and this red stains CD3. So with a polyclonal process, you'll have a mixture around 50% of each of red and brown, versus a monoclonal pattern will be greater than 80% of either red or brown. And in our study, all cases of verified T-cell lymphomas and cytology specimens showed a monoclonal process compared to our controls. There are a few caveats of using this dual stain. TRBC1 is only expressed in alpha beta T cells, so a gamma delta T cell process wouldn't express TRBC1, but if you see all red, that also could be very suspicious for a gamma delta T cell process potentially. It's not helpful for detecting subpopulations. It needs to be majority to be able to interpret the stain. The sample also needs to be sufficiently cellular. So in conclusion, this TRBC1 CD3 IHC could be really helpful for you to bring on in your lab as a surrogate for TCR clonality. You can read all about this study and get more details about the IHC protocol in our publication on the Cancer Cytopathology website.